Okay, let's talk about the composition of a protein. These are the last sets of slides in our note packet, starting at slide 16. Amino acids are combined in a dehydration synthesis reaction, forming a long chain of polypeptides. A polypeptide chain can usually be between 1 and 300 amino acids long. So those amino acids are linked in this long chain, and the chain doesn't stay the same shape. All right, so proteins are shape specific. The shape of protein includes primary, secondary, tertiary, and quaternary structures. And this is best shown using a picture because shape determines the function of a protein. In this particular case, the primary structure is shown as a long chain of amino acids. Each circle or sphere here represents an amino acid. That amino acid chain will fold on itself because it's so long, and because those molecules are floppy, they will interact with each other. So the different amino acids will have attractions and connections to other amino acids, creating a secondary structure shown in this picture, showing the folds. Now those folds will also fold over on each other, creating a complex tertiary structure, right, that gives the shape for the molecule. Now the last and most complex level of protein shape is the quaternary structure. And quaternary structure is indicated by the two different colors here representing two different proteins. So different proteins can join together to form a larger protein structure, and that protein will have a particular job. So quaternary structure is determined by multiple proteins interacting together to do a job. Which brings me to enzymes. Enzymes are the biological catalysts of your body. They speed up chemical reactions, and they are not changed in the reaction. And because they are not changed in the reaction, they can be reused over and over and over again. They lower the amount of energy needed to make a chemical reaction happen. And they are enzyme specific. So the enzymes will only work in specific chemical reactions. We refer to this model as the lock and key model. Enzyme names are typically based on the reaction that it catalyzes. So enzyme names will often end in ACE, that's dash ASE, so lipase or protease, and the name indicates what it's reacting with. Enzymes are typically affected by pH. If you change how acidic or basic an environment is, it can affect the activity of an enzyme. It can also be affected by the temperature. If you raise the temperature up too high, then the enzyme will change shape. There's a very important term for this change in shape that you need to be familiar with, and that term is denaturing. Right. If an enzyme changes shape and it won't work the same anymore, that is called denaturing. It's denaturing the protein. All right. You can also cause enzymes to get their jobs done faster by increasing their concentration. So if you were to think of, okay, if I need to unlock a door, the more keys I have, the faster I'm going to be able to unlock the doors. That's essentially the idea of concentration. More enzyme means more work is going to get done. Then that brings me to the last of our organic molecules. All right, the organic molecules that we're going to address now are nucleic acids. Nucleic acids include DNA and RNA. These are not workers per se. DNA is a storage molecule. It stores genetic information. And the molecules themselves are made up of nucleotides. And I have a picture of a nucleotide on the next page. But DNA and RNA have their own nucleotide structures. They're very similar, but there are specific differences. RNA uses ribose as its sugar backbone. DNA uses deoxyribose as its sugar backbone. That is the name part that you should be familiar with, because RNA has ribose and DNA has deoxyribose. So you have the sugar that we just identified. There's going to be a phosphate group, and then the last piece, which is the actual code, is the nitrogen. These are nitrogenous bases. And when I go to my next page, we'll see how these are actually going to look. So this is the nucleotide structure. A nucleotide is made up of a phosphate, the red dot, a sugar, in this case it's DNA sugar, this is deoxyribose, and a nitrogenous base. These are connected together by bonds. So the bonds will join one nucleotide to the next nucleotide, giving us a long strand. Now, DNA is two-stranded. So looking at this, the DNA molecule is a double helix. It's two strands joined together in a curling, kind of a twisted ladder shape. And those nucleotides are shown by the colored bases and the ribbon edge. That ribbon edge is the sugar and the phosphate that we saw in the previous slide. So if I go back here, here's the sugar and the phosphate zigzagging back and forth. That would form the ribbon or the edge of that ladder. The steps of the ladder, of that twisted ladder, are the nitrogenous bases. Those come in different varieties. The nitrogenous bases in DNA include adenine, thymine, guanine, and cytosine. We abbreviate those A, T, G, and C. And we can see that right down here. In this part, in the DNA molecule, oops, I got a little extra one in here. Let's get that out of there. All right. The DNA molecule has adenine, thymine, guanine, and cytosine, A, T, G, and C. An RNA molecule substitutes and will never have thymine in it. It's going to have adenine, cytosine, guanine, and uracil. And again, these bases are just the steps of the ladder. The sugar and phosphate are going to repeat just to make the structural backbone. RNA is single-stranded, and we see that in this particular image. DNA is double-stranded. DNA is always stored inside the nucleus of a cell, and RNA is a working copy of that DNA molecule. The DNA molecule is capable of being separated in between the two strands like Velcro. It can be detached and reattached in order to be able to copy the code. So by unzipping it like a zipper, I can make a single-stranded copy of the DNA code that can go and do a job. And this is why RNA is so important, because the RNA will be able to leave the nucleus and go help create the proteins that the DNA is designed to code for. So finally, this summarizes the actual message here. DNA contains the genetic information to construct proteins, but RNA is responsible for actually carrying that message from the DNA to direct the manufacturing of proteins inside the cells of your body. So I'm going to quickly go to an image. All right, if I look right here, I can draw for you how this kind of looks. The DNA code is a double strand that can be separated. I can unzip and separate those molecules, the long strands of the DNA code. So I'm going to have A, T's, G's, and C's in here, but I can separate them. G, C, A, T. Right? And in fact, it can be T and A as well here. Right? So this represents the code itself in the middle. If I unzip this, I can make a temporary copy and you'll notice if I pull this copy out in a way, it becomes G, C, A, and U. And now I mentioned uracil is only found in RNA. And it's substituted for thymine. So if you ever see a genetic code with a U in it, you know that it's RNA. If you ever see a genetic code with a T in it, you'll know it is DNA. Right? So those are very important facts. Now this RNA code would travel out of the nucleus and it would go help to make a protein. And those proteins do the jobs inside your cells. Those proteins could be structure of muscle. You can make more muscle fibers. It can be hormones that you're making. It could be connective tissue like cartilage or keratin for making hair. This is all made with proteins. They do jobs inside your body. Your digestive enzymes are proteins that digest your foods. And as we learned in the protein section, right, enzymes are special types of biological proteins called organic catalysts. And they do specific jobs like salivary amylase. If it ends in ACE, it's an enzyme. So this is an enzyme that's found in your saliva. It helps you to break down carbohydrates, right? And the carbohydrate that it breaks down is called amylose. And anything that ends in ose, we learned earlier in our unit, is a sugar. So we've got a lot of different ideas coming together here that you need to be familiar with. So even though we're talking about DNA and RNA here, it refers back to our learning about proteins and enzymes, and it also refers back to our learning about carbohydrates and how to know what different molecules are made of. All right, well, hopefully that helped, and if you have any questions, come and see us on our Wednesday office hours. Take care.